Oh my, that was abrupt again. Hi, boys and girls. So, oh my, I'm 26,333 days old today, which is half of Satan. I'm half Satan today.
Wow.
Half Satan. And I've gone through 1/3 of my 26,000th days.
Actually, I need to clarify, you're half the Antichrist.
Half the Antichrist.
Yeah.
That's right. Mark of the Beast is the Antichrist.
Yep. Which is kind of cool, I guess. I don't know. Trying to figure it out.
It makes a good T-shirt. It really does.
Does that mean you have a birthmark on your head, kind of like Damien from? I don't know if you've seen the.
I have one on the left side of my nose. It was really bright when I was a child, and a teenager. My mom told me it was to keep me from living a life of crime because they'd be able to identify me by my birthmark. So instead of the mark of Satan, it was the mark of can't do crime, I guess, for my mom.
You know, speaking of the forehead and everything, for some reason, when people get on YouTube or some other video and the camera comes up close, I wonder what percentage of people have this little gap in one of their eyebrows where they, had a cut or a scar and the hair has never grown back since then.
There's a few.
A lot. But I think it's a common injury.
Yeah. It's like, I keep seeing this, and it's like seeing a car that's just like your car all the time. Or it's just like, like let's say you have a crush on somebody. You always see their car. Or something, and it's just like I always see that, and it just bugs me. I don't know why.
It's a common injury, and it's because our nervous systems are pretty well suited to induce an instinct to rapidly turn our head when things are coming directly towards our eyes to protect our eyes. So it's very common to get injuries right around the eye. To protect us from that, and it's something that happens super duper quickly. We're not aware that we're doing it. I also have a scar very close to my eye. It's a tear scar, so it's not on my eyebrow, fortunately, but it's similar. So a lot of people have those injuries because of that basic phenomenon of just protecting the eye. Cool. It's kind of like the what is that phenomenon with the World War II, the B-52s that were coming in, and they're looking at the damage that was on the B-52s that would come back from missions, and they would see like some areas that weren't damaged in these, and they're like, well, do we need to protect the areas that are getting hit? Like, no, actually, we need to protect the areas that aren't getting hit. So it's a similar thing. Like the eye, you look at the damage of the eye around the eye, because that's indicative that we have these mechanisms that protect our eyes from actually getting hurt.
Wow.
Yeah.
And leave it to Dr. Chris to give us a great neurological explanation for a phenomenon. And that's why we need more Dr. Chris on Answers in Atheism.
I have a story. I talk out of like one side of my mouth, sort of, and it's one of the reasons I have trouble enunciating, I think. And I think the reason for it was when I was like 11 years old, I was chasing two girls across a pasture. Something we always did on the Iron Range of Minnesota is chase girls across the pasture. And there was a 12-foot section of fence that somebody forgot to take down with a barbed wire right at my mouth level, and I hit that at a full run. And I bounced off it, and I had to eat through a straw for 2 weeks. And it was
that's rough.
But I have a scar, and it's something that I remember, and I think it had something to do with the way I talked, because there's something wrong with that one side there. There's nerve damage and stuff. It was quite a serious injury, but I never caught the girls.
Because
Got
that's
away.
what you do.
For the rest of my life, I never caught one. Damn it.
Blame it on the barbed wire.
It was a barbed wire. Yeah, well, I didn't chase them so fast after that. I tried to slow down a little bit. Slow my roll. Anyway, so okay, we have Kent Hovind humiliated in impromptu debate on flood geology in the Grand Canyon, which is a little video, and then we talk a little bit about collagen evolution.
Yeah, let's talk about your stuff first in case Grayson shows up, or we can talk about without him anyway. Yeah.
If a person is incapable of feeling humiliated, should we go on and say that we humiliated them?
That's true.
I don't know, because and that's a good link. It's good clickbait. It's good leading way to get you to click on a link.
Well,
but,
we could say he was destroyed, right? Like the
Yeah.
Ben Shapiro style
Destroyed.
of
Yeah,
putting
that's
it
a
all in
better word.
caps. Yeah.
Well, the most humiliating thing has got to be when you're humiliated and you don't know it. You're the only one who
Yeah, that's true. Yeah.
It's humiliating. So I'm not sure if that's it or not. But let's see. What do we got here? Let's see if I turn the camera on.
Collagen.
So a large number of collagen-like proteins have been identified in bacteria during the past 10 years.
Mm-hmm.
So they used to think prior to like about 2010, I've got another paper here that says something on the order of this shows that, collagen is unique to metazoans and basically eukarya, and that's where they thought they existed. But now I forget what it is exactly. Let me put that paper a little bit bigger and see what I can glean here in the end of it. So down at the conclusion, and let's see, where is my conclusion? There it is. Okay, hundreds of protein sequences containing GXY, basically, or glycine and any two amino acids domains have been found in bacterial genomic databases, and 8 of these proteins coming from both pathogenic and non-pathogenic bacteria have been expressed in recombinant proteins in E. coli and characterized in detail. It's been shown that all the predicted collagen-like structures do form stable triple helices in bacteria, with protease resistance and melting temperatures similar to animal collagen. This suggests that most, if not all, of the GXY regions of sufficient length in bacteria proteins are likely to be triple helical. And surprisingly, that they may have a thermal stability in the area of 35 to 38 C range, which is highly stable. And they do this without the hydroxylation of the proline. Like the most common motif in collagen in vertebrates is a G proline, so glycine proline. And I'm surprised that Sal Cordova, when he shows his diagram, is in that paper that he just posted under Lynchpin's video, when he shows that, he notices a lot of patterns, but the pattern he doesn't talk about is that there's a lot of GPPs sprinkled throughout, and there's a lot of GXP and GPX or GPY. So it looks like the original metazoan source of that particular motif was a GPP. And then later, and then there's no problem with later having hydroxylation of the proline evolving to make it even more stable and to make it more powerful in metazoans, in vertebrates, because we need a lot more. I mean, bacteria only needed to stick to a surface, stick to another bacteria, or stick to each other. And they apparently use collagen to do that. So lo and behold, the more we learn from science, the less likely it looks that these things did not evolve. You know how the creationists love to say,
that's the way you phrased it. Yeah.
the more we're learning, the more it looks like God did it. It's like, nope, not. And then the other thing is Cordova does a calculation in his paper that there's like 1 in 16 probability of it being a glycine, right? And then he goes on to extend that to all 23 GXY domains. And he comes up with an improbability of 10 to the 337th in order to evolve, right? Assuming that this whole protein had to instantly form with 23 GXY motifs, right? Which is not the case. These bacteria, some of them have 6, and they form triple helices.
Yep, exactly. Yeah. And that you could easily imagine a duplication of portion of a gene that now doubles it to 12. And then you can double it again and not lose some. And then you can finally create like the superstructure where you have a repeated motif over and over. It's a relatively straightforward, natural pathway that can explain where this repeated motif can kind of come about.
Right. So,
like Lego blocks almost. Sorry, go ahead.
How do you gentlemen account for the fact that he does this calculation but doesn't seem to take into account those other processes?
I don't know.
What accounts for that?
Well, that's him creating a straw man. He's calculating the tornado in the junkyard scenario.
Yeah.
That a tornado comes through a junkyard and assembles a Boeing 747. And so a completely random process resulting in something relatively complex. Well, clearly that the odds of that happening are longer than the span of the universe. But we know that like evolution of proteins, first of all, they're not necessarily specified, not like so in the Dembskian sense that these need to be highly specified. You have to have this precise sequence, otherwise it completely doesn't function. There's a lot more flexibility about it. But just as you're pointing out, that like we can have smaller domains. You don't even need the whole thing. 6 is enough.
Right.
Yep. To get some function, doing essentially the same thing.
The fact that there's so many
they often bring up, you're making up stories of how it evolved, but like if you take into account co-option and how it might have had a different function in the past, then that could explain part of that as well.
Sure. Yep. Well, and that's the thing about the family of collagens. They're quite varied, actually. There's 28 of them in mammals, and they kind of fall into these subcategories that do different things. They are the most abundant proteins that you can find in an animal, but these different categories do different things, and so like and you can compare like the phylogeny of the family of collagens in the way that I did for the potassium ion channel for Sal in our conversation where he suddenly had a meeting at the very end. Meetings come up sometimes, but you can do the same thing for collagen. And the thing is, like the collagens are much more varied in their function than potassium ion channels. And potassium ion channels are quite varied, but they all kind of regulate potassium ion flow through the cell membrane. Collagens do wildly different things, but they're all collagens.
That's the other thing. Yeah. And even in bacteria. But this paper from 2002 said, in this respect, collagens and collagen-like proteins are unique and essential to the appearance and diversity of metazoans. And it turns out by 2010, they started discovering collagen-like proteins in prokaryotes.
It doesn't surprise me, but that makes a lot of, I didn't know that. It makes a lot of sense that you would end up finding it in prokaryotes, given that this is clearly a family of proteins that's been around for a very, very long time, because you find this family in all animals.
Yeah. Yep.
Invertebrates, vertebrates, all kinds. Yep.
Yeah, they talk about duplication in this paper called The Evolution of Collagens. I wonder if he ever typed that into Google and looked it up.
Oh, I'm sure he did. He must have, and then just ignored this paper, I'm sure.
Ignored it, all right.
He couldn't quote mine anything out of it, I bet.
Yeah.
So no helpful Jerry Coyne quotes in this one.
Yep.
Speaking of Sal Cordova, didn't you have a discussion with him about genetic entropy once, Dr. Thompson, or am I thinking of
Yes.
someone else?
I did. Yes.
Ah.
I went about as well as you might think. There's still more I got to cut from that one. I put some of that up in videos. So when I recorded the video, like the audio wasn't great, so I kind of took it down and I've been editing it. He has that whole thing on one of his channels because I let him have the file. So it can be seen there. And we got into genetic entropy and the conversation. We basically, I got him to talk about creationist genetics, and because one thing when he approaches this topic, we're talking about collagen because he wants to talk about collagen, right? Because he thinks that this is a big problem for evolution. He doesn't ever want to talk about the details of his model, right? Like, how did Jesus make all the different collagens? We should know that, right? When did this happen? Why did he make such similar collagens in, say, chimpanzees and humans? And why are they also sort of similar to mice? And then why are they still kind of similar to chickens? But then you look at sponges, now they're different, but they still have some. And now you got collagens and bacteria. So why is he using the same? Why did Jesus put so much of the same thing, but change it in just the right way that would match exactly if all animals descended from a common ancestor?
It was his creative diversity.
Mm-hmm. Yes, I suppose. Anyway,
if you look at the collagens, the glycine part, the G, is highly conserved, but the other part, the XY, basically you can see all sorts of phylogenetic
variation.
In that, and it's amazing.
Right. Yeah, because that glycine is the thing that's key to creating that 3D structure the collagens have, the helix.
Yep.
But the other parts aren't, they're more flexible, so you can get evolution there. And as you said, you compare it genetically, you compare the chimpanzee collagen 1A to the human collagen 1A. They're very, very similar. Those spaces in between the glycines, a lot of those are exactly the same between chimpanzees and humans. But then you compare humans to birds, now you see some differences, and then you compare them to I don't know, Drosophila, and then now Drosophila, all of a sudden there's more differences.
Sal said some pretty incredibly ignorant things about in the Grinch Lynchpin video that he did. I was absolutely amazed. Let's see if I can find one of them. Okay.
If I may say one, the genetic entropy thing. Does anyone know who Zach Hancock is on YouTube?
No,
I do not.
I think he is a biologist. I've talked to him a few times. He recently, he's doing more videos nowadays. And one of the more recent ones is actually talking about the fundamental flaws with genetic entropy. So if anyone hasn't seen that yet, I do recommend everyone go subscribe and watch that, because he does a really good job. And at the end, he talks about how he did a simulation of all the things Sanford could have done but didn't, or kind of giving it the best shots for, and how it still ended up with no genetic turning out. But I'll leave the details of that to the video.
What's his name again?
Zach Hancock. Zach B. Hancock, I believe. Yeah.
Okay.
That's the name of his channel too, I think.
Yep. So in the Grinch Lynchpin video, and I can't find it, what he said is basically how could, if collagen evolved, how could the animal live? Because it would have scurvy. Because scurvy is what happens when you, like, vitamin C is responsible for hydroxylating that proline and making our tendons in big, large vertebrates, right, much stronger. And if you don't have that, basically you can fall apart and die. But that was his idea, that you needed the vitamin C and you needed the proline, the hydroxyprolinase, I think it is, a certain enzyme. And no, you don't. These bacteria do not require that hydroxylation. And these things still form triple helices, and they're still sticky, and they still bind to fibronectin, which is a protein in our extracellular matrix, right? So you don't need any of that stuff. And he just says, oh, the bacteria would get scurvy if it didn't have vitamin C, right? It's like, no, Sal, no, no. I couldn't believe he said that out loud. Oh my goodness.
Scurvy dog bacteria.
Yeah.
But now, what I want to do,
chocolate, they'll get acne too.
That's right.
Yeah,
but only in their adolescence.
Yeah, and I downloaded a couple of genomes in full, and I want to start looking into the exact nucleic acid sequence in these collagens across various species, and I want to see how all that hashes out, so that I can counter some of the other bullshit that he says. And some of it is in this paper. You notice that his other authors did not give him permission to use their names. I'm pretty sure it's Sanford, Baumgartner, or whatever, and or whatever, the usual suspects, right?
Right.
Anyway, so we use 4 very well-studied proteins to illustrate. Okay, first of all, principles for evaluating hypotheses of de novo origination of proteins via frame shift mutations. Okay, and he keeps talking about it like evolutionists think that frame shift is a major way that evolution happened. And to my knowledge, that's not the case. Frame shift is...
Most frame shift mutations are bad.
Right. Very bad, yeah. But now frame shift mutations, if they happen on the C terminal or the end of the protein, right? If they happen toward the end, oftentimes the protein is still functional. So a frame shift can make it through the filter of purifying natural selection if it happens toward the end. But I also think if it happens somewhere in an exon, that the intron is still capable of basically going to the next exon, and picking up the 3 sequence again. And there's some evidence of that. I found a paper a couple days ago that says that what will happen if there's a frame shift in that exon is oftentimes that will be skipped, and then the new protein, if it survives, will have that exon as part of its intron, but it'll still splice normally and form a protein that could be functional or neutral or whatever. So I mean, there's the frame shifts are not like total death, even if they happen somewhere earlier in the protein. Yeah. There's the variations. It's just another means of evolution, another means of mutation, which is the raw material for evolution, right?
Okay, wait a sec. In 1984, Ono advanced the theory that frameshift mutations. Didn't he advance the idea of gene duplications being important? I don't recall him saying anything about frameshift mutation.
His book is on gene duplication, but for this one protein, nylonase, the nylon dissolving thing they found, he found a way that a frame shift could have made that happen.
Oh, okay, I see.
Later on, other researchers found existing, bacteria, prokaryotes, that had that protein in them already anyway, and it just happened to work for nylon. And him and Sanford, I think, wrote a couple of papers about 5-6 years ago, and they got them published somehow about nylonase, and they're sort of overturning Ono's hypothesis. Well, Ono's hypothesis was overturned 15 years before that by another researcher. So I don't know what their paper was for, because it had already been, they already started to cast doubt on it. But yeah, Ono wrote a book on gene duplication as a means of evolution, which was, I got it somewhere on the shelf. Anyway. But anyway, so we use 4 very well-studied proteins to illustrate the many levels of constraint. In other words, the many reasons why frameshift mutations will not result in anything positive that would make de novo beneficial frameshift mutations. Very unlikely. But these should be very rare and should be. There are doubtless frameshift mutations that do give rise to beneficial new proteins. There he is. But these are very rare. Yep. The whole thing is rare. Evolution is beneficial mutations are rare. That's why there's lots and lots of life. That's why we have huge populations, right?
Mm-hmm. Right.
Anyway, but then he goes ahead and he picks 4 proteins that he states that everybody knows that they did not evolve by frameshift mutations, right? They were like duplications, zinc fingers and collagen are 2 of his examples. And then he goes ahead and proves that they shouldn't or couldn't have evolved that way for a number of reasons. And the whole thing is, I thought it was pretty amateur. I don't know. Did you read it or did you look at the whole thing?
I glanced over it. I would agree. It's pretty amateur. I mean, it's not written terribly well. It's okay, but like, there's a lot of redundancy. The big glaring thing for me is that it's just a straw man. I mean, he's talking about a phenomenon of a mechanism that can lead to change in the genome, but generally tends to be pretty deleterious. So this isn't going to necessarily be like the main way by which many proteins are going to originate or evolve. And yeah, so it's largely just a straw man. So
yeah.
And he makes it clear, because he said every single time that he talks about a protein, and correct me if I'm wrong, like, because I counted only 3, even though he's talking about 4, but there's only 3 proteins that he discusses at length. And every single one of them, he says, like, well, no one ever is claiming that these evolved by frameshift, but I'm going to why don't we pretend that that's the case and talk about why that's impossible?
Right.
Okay.
Oh, what he meant to say, okay, this paper is a work in progress. So he hasn't completed it. I think he meant they found 4 theoretical constraints.
I see.
Okay, not and he uses 3 proteins.
Not 4 example proteins, because I thought he did say someplace that he has 4 example proteins that he talked about.
There's a couple of typos in it and stuff. So
yeah, and that's fine. I mean, geez,
he did point out that this is nowhere near done yet.
Sure.
It's in the early stages. So.
Fair enough.
Yep. So where does he come up with this 337?
I have a quick question. Okay, he says it's not near being done, but does that mean he's going to change his argumentation? Because this is what's important, is the direction of his argument, and he should have that pretty much formed, shouldn't he?
Yeah.
Yeah.
So, but when he's using a straw man, it's not like he's going to eliminate that straw man.
Not off to a good start.
Yeah.
He's going to have to eliminate almost everything in this paper.
Right. Yeah. Eliminate anything that is not a fallacy or a just an error, and what's left.
Yeah. Oh, let's see. Where is it here? Okay. Here's where he does the odds of glycine appearing is roughly 1 in 16, right? And then under the assumption of equal probability, the odds is whatever. So then he multiplies it by all 23 GXY domains in that collagen A1 protein, call A1, and he comes up with this 10 to the 406 improbability, and it violates the law of large numbers. So that's what he says.
Mm-hmm.
Anyway,
sure.
Which is ridiculous, because we know that shift duplication can happen. Once you end up with 2 of those things, ending up with 4 and 8 and whatever, or when you have 3 or 6, they will end up getting duplicated. And in fact, I think if you looked across humans, you'd find all kinds of variation. There are some diseases that are a result of too much duplication, but it happens.
Sure,
we know
it does happen. Now, and as I said, I don't think that frameshift mutation is necessarily like the major source for genetic variability that can be useful to natural selection. but there are examples. I don't think in the paper he wrestles with any of that.
Right.
There's a great paper that I put in the private chat. It's from BMC Ecology and Evolution, and they talk about using comparative genomics to identify a frame shift mutation that must have occurred in plants, and it's in a particular like a MADS box gene. So this is like probably some kind of transcriptional regulator.
Yeah.
And they find a lineage where they can see a frame shift mutation that's occurred compared to the other plants that are outside of that branch in which it occurred. And clearly it got established. And they have a whole section in the discussion about hopeful monsters, which is a nice discussion of the fact that like you don't necessarily expect to see this happening too often, but it seems like hopeful monsters can occur. So the idea of a hopeful monster is like most mutations are going to result in monsters that are not hopeful. They just are going to be deleterious and killed off, right? Or not even be born, or not, if these are plants, they're not going to, have flowers or petals or whatever, right? So but sometimes a fortuitous mutation can result in something that actually can be beneficial to the lineage. And this is an important gene. So the fact that there was this frame shift mutation that occurred and that was like rooted in, relatively back in the tree, clearly it had some benefit, and it didn't result in something that was utterly deleterious. So
if you get a chance, send me a link to.
I put it in the private chat. It's the very first link in the private chat.
Yeah, Speed, you've got several links here in private chat to put over there into the description.
Oh my goodness, you guys!
There's only 3, and one of them is a YouTube link.
You think this is all I have to do?
I know, right? We're hijacking your stream now, but at least this one is relevant to what we're talking about.
Yeah, the one I contributed is basically Religion for Breakfast did a really good video on what yin and yang really is.
Okay.
The whole concept, but I thought it's pretty good. I think people might appreciate it.
Yeah, cool. I like the yang. We'll have to get James a person, a generic James person, to come on and talk to us about that.
That's a good idea.
Yin and yang. So okay, then this is Cell's picture, that he keeps showing of this repeating pattern. You can see that those G's are highly conserved.
Right.
Across all lineage. But those other two, X and Y, they're not. But then if you look at them, it's glaringly obvious that there's a lot of GPPs in there.
Mm-hmm.
I don't know. I can't highlight them. So, and there's a lot of GP, GPY and GXP.
GP
over the place. It looks like GPP was the winner in the beginning,
right?
Or one that it all came from. But of course, you have to do a lot of analysis, and that's something I'd like to do, is look into a little bit of the collagen.
Are these the amino acid acronyms?
Yeah.
Yes.
Yeah.
The 20 amino acids used to make proteins, they all get a letter.
Yeah.
Yep.
Okay.
It's frustrating because the letter doesn't necessarily match the first letter of that protein.
Right.
I often look at the table.
Yeah, it's kind of cool because like aspartic acid is spelt with a T, aspartic acid.
Yeah.
But it sounds like aspartic, so they use a D. They do shit like that, which is kind of cool. I don't know. It's like they have a sense of humor.
I think it's frustrating.
What's that?
I think it's very frustrating.
It is.
I constantly have to consult tables when I look at amino acid strings.
Instead of glutamine, you have to say glutamine with a Q, right? So you just have to learn to pronounce them weird, and then you're fine.
I suppose, yeah.
I still stumble occasionally on some of them. It's like, wait a minute, what is that one again? And I second guess myself. And I love V because it's valine. It's the only goddamn V, and I like that one.
Yeah, that one's nice.
That one's nice. And G, glycine. Yeah, glycine's nice. What the hell do we need those other 18 for? Anyway. So the other things is he's okay. There's basically the postal code that he talks about is how it ends up getting delivered to the endoplasmic reticulum and to the Golgi apparatus, and ends up getting basically on the outside of the cell. So it has a postal code, and those things they found in bacteria, in prokaryotes. They found many sequences that are homologous with that. And then you have this NCI domain, which is very helpful in how the triple helix forms. And I have a paper here on that too, as well. And I haven't looked into it. The thing is, when you look into this shit, you can find very, very quick and very credible pathways for all the evolution of any protein that we have extant, right? So anyway, I'm done. I'm done.
I like this comment by Brian.
Quotamine is a
wordplay.
Oh, quotamine. Oh
Nice.
my god. Oh my god, I gotta clip that. Yeah, thank you.
Can you bring up that link that I sent in the private chat? Is that possible? Because I know I don't quite have access on Restream to share, because otherwise I would do it here. But
was that a link?
there was a link. It's BMC Ecology Evolution. It's in the private chat.
Yeah,
Scott, you see it, I think, right?
A simplified explanation.
Yes, I saw it.
Yeah.
You saw the link?
It's
The
go
very
up.
first one. Very
So-
first one.
Right. I have the title separate from the link.
What was that? Did you hear
I
that?
don't know. Yeah, that
Oh,
was.
I was the
There's
only one.
something, there's something around here on my desk, or maybe it's my laptop that's beeping, but I cannot localize it. But it's driving me crazy,
Okay.
and I do apologize.
Okay, let's see. I got to copy this link. Let me see. Put that in my description. I didn't see the
Is
link.
the Madsbox mutation you were talking about?
Yes, that's right.
Okay.
And in eudicots, which are, I don't know my plants.
They're
I'm
a type of
neuroscientist.
angiosperm.
They don't have
flower
neurons. Yes, it's a clade of flowering plants having 2 seed leaves upon germination.
It used to be monocots and dicots, but then they realized dicots isn't a monophyletic group, so to speak. So now it's just eudicots and some other weird things.
Right. So there's
And
quite a
little
few
cots,
different
you know.
eudicots. Eudicots include sunflowers, dandelions, cabbage, apples. Yeah, it's a bunch of things. Okay. All right. But this paper, if you go to figure 2, just kind of scroll down. So, they've looked at this gene in a bunch of different organisms, and that's where it shows the frame shift event that occurred. And so all the, organisms below that have a frame shift mutation that they then show in figure 3 what that frame shift looks like, I think. I mean, I'm only glancing at this during the stream, so I don't know for sure. But in figure 3, they show examples of like what that theoretical frame shift would have looked like. And they can show it in each of these organisms that are in that particular clade. And that there's just a shift of like one amino acid that resulted in a change of all the proteins that would have been in that clade. And they line up so there must have been a frame shift mutation that occurred many millions of years ago in that group. And then it got rooted, so to speak. I'm pretty good with puns, I guess. I didn't even mean to do that, but it got rooted into the genome, into these plants that right there. So all the organisms that would have been descended from that common ancestor when the frame shift mutation occurred, they are there. And obviously it wasn't deleterious. And so then at the very end of this paper, right after figure 6, is a part where they talk about molecular hopeful monsters.
Yeah.
Right there. Yep. And it looks like a nice discussion. I haven't read through the whole thing, obviously, because I'm just reading it right now. But
Good.
I'd be willing to bet that while hopeful monsters may occur every once in a while, I still think slight variations are the norm. at least some of the time.
I think that's right, and that's basically what the discussion is. But obviously, this paper is showing that frame shift mutations can be, sort of again rooted into the genome, and but I do think that they're rare. I would totally agree. I'm sure that this is not
when you have genetic mutations that can cause all variation, there are always exceptions, and
exactly.
There are always different sources of variation.
Of course, yep. And I think single nucleotide polymorphisms are going to be the most common form by far. But there's other examples of mutations that can occur. Obviously, genome duplication is a huge event, and that tends to be rare. But we know of plenty of examples where organisms have
or even whole genome duplication is even rare, but it's happened a lot
chromosome
in
duplication or whole genome, exactly.
Yeah.
And the species I work with, Xenopus laevis, that's my icon. That's a tadpole head there that I use as my icon. That species is tetraploid. It has 4 copies. It has a sister species called Xenopus tropicalis that has half the number of chromosomes. And we know, you can do molecular dating and find out, when was the last common ancestor between them? And it was only around 10 million years ago. So Xenopus laevis underwent a whole genome duplication very recently, and that was not present in the other ancestors. And Xenopus as a whole group, there's other weird duplications that have occurred in other sort of distantly related species too. So I don't know why that group is prone to getting genome duplications, but apparently they are. I mean, you see this also in fish, and plenty of fish also have many copies of genomes. They've undergone multiple duplications.
According to Kent Hovind, they now have different numbers of chromosomes, so they must not be related, according to evolutionists.
Even though you can look at both chromosomes and see that there are 4 copies, there's 2 of them that are more similar to each other than the other 2, but they both are clearly copies of each other. There's 4 of them. That's why we can call them tetraploid. Yep. And they have double the number of chromosomes of their sister species. Yep. I know.
Yeah, but it doesn't generate new information now, does it?
No, clearly not. You're just duplicating the information.
Right. Like a book.
All the specified complex information that, the Holy Ghost wrote into some ancestor frog species when it was created on Creation Day. Yes.
Fish, and then you have fishish. Which could be a word. I don't know. Some people look at fish who's that? Oh God, I can't think of the politician because I don't keep up. But no, he looks like a turtle. What the hell's his name?
McConnell, Mitch McConnell.
Yeah, exactly. He looks a little fishish too. I don't know. So, yeah, fishish.
Definitely turtle-ish.
I'm going to stick with it.
This may be a little off topic, but before we move on, Dr. Thompson, do you happen to have any thoughts on the whole dinosaur soft tissue thing, or is that not really in your? What are your thoughts?
Okay, so it's certainly not something that I'm expert in, but I have done some reading up about it, and I've seen some of this talked about in videos and things. And then I think it was Gutsick Gibbon that actually had a really great video talking about it. So it is true that you can find quote unquote soft tissue in some dinosaur bones. But the soft tissue, quote unquote, is not what we think of as soft tissue. My body is filled with soft tissue. I have too much soft tissue in my body, outside of my bones and inside my bones. But obviously when you're left with a bone, all you're left with is the bone. And by soft tissue, what they mean is that there is proteins that you can find inside these bones. And in fact, actually, some of them are collagens. Collagens are highly stable. They're incredibly stable. They can last for a very, very, very long time. DNA is surprisingly stable, but it has a half-life that will, lead to decay of the DNA over tens of thousands of years, out to maybe 100 or maybe 200,000 years. We have ways to kind of refix it all and sort of compare across multiple strands of DNA to get a genome when it's really degraded. But you're not going to find DNA in a bone that's 65 million years old. But some proteins have state stability out to that length of time.
Right.
So they're incredibly stable. Collagens are one of them, but there's only like 6. So when they talk about soft tissue, all you're finding is remnants of bits of collagen and a few other
typically
proteins
fragments.
inside the bone, and that's it. Yeah, you're not actually like opening up the fossilized bone and it's like blood and like
out.
yeah, it's not nothing like that at all. Of course not. And
Well,
actually it's evidence that okay, we don't find, so collagen does have like a degradation half-life, and getting up to like late dinosaurs and that died in the Cretaceous, that's when you would expect to see still some collagen and a few other proteins. But when you look in bones that are from, say, 300 million years ago, you don't expect to see any collagens. And in fact, people have looked and you don't see any because they will have degraded too much. So
Right.
if all animals were killed in the flood 4,000 years ago,
should be all over the
and
place.
obviously dinosaurs still have soft tissue in them, shouldn't some of the animals like Ichthyostega that lived around 380 million years ago also have soft tissue? But they don't. So in fact, this whole soft tissue argument actually is a nice measurement of the fact that the Earth is really, really, really old. Some animals died tens of millions of years ago, and then others died hundreds of million years ago.
Because I bring this up, like, why do you think it's not, I use the word sensationalized, but I should have used journalized or media-based or whatnot. But like, why do you think it is that way? Because these findings are so rare in respect to all the hard parts we typically find. And in fact, we brought up what you said to one creationist about, well, we usually only find the proteins or at most the protein fragments. They said, well, that still shouldn't exist, even though that's the most stable part of it, typically.
Right. Those are the most stable proteins, and the best that we can tell is that they would last millions of years.
Right.
So it's perfectly consistent with that. Like, you can find bones that are maybe thousands of years old, and you will find a lot of other proteins other than like this like 5 or 6 that you can find in the soft tissue dinosaurs.
Right. And I also bring up the point, like, when you talk about like fossilized skin, for example, it's not really skin anymore. Like when you have a frozen mammoth carcass, for example, it's not skin in the same sense. It's more like a remnant.
No, that's not true with mammoths. If they've been frozen, it's not skin in like our skin, or say from even an elephant skin today. Obviously, it's highly frozen, fractured, and degraded, and maybe decomposed to some degree. But there's still a fair amount of like actual biological tissue that you can get out of a mammoth that was frozen like 5,000 to 10,000 years ago. And that's why we can have the mammoth genome, and there's talk of like making new mammoths.
When you talk about like hadrosaur skin on a dinosaur, that's not skin anymore.
It's not skin. That is highly mineralized. The same goes for like fish skin and any kind of skin or soft part that happened to be fossilized. It's still mineralized. It's entirely mineralized. There may be remnants of chemical traces that give us some hint of like what kind of chemicals might have been in the biological tissue, but it's from the process of fossilization and mineralization that, well, just as like a geode, as it forms, the colors that it takes on might be because of some of the trace elements that are being seeped into the geode. The same would be for a fossil. Like, some of the chemical that made up the organism that died millions and millions of years ago, those chemicals are still there, and they're going to change the mineralization that occurs right there. And some of that can include soft tissue under particular mineral fossilization conditions.
Like when you're talking about these stable fragments, it's a similar question to asking, okay, you have bones. Why aren't all the bones degrading over time? Well, because essentially bone is calcium carbonate and all the other minerals associated with that. And what is that? Rock, basically. There's no reason
and they become mineralized over time. It's most often the case they become mineralized, so they get filled in with silicon oxide or other minerals to then like make a permanent cast or an impression of the specimen itself, and of course it'll be incredibly detailed, but it's been mineralized.
Right. And so far, like you said, we haven't even been talking about the environments that could have been preserved in. Maybe like I think the really good examples we found have been in river systems or some kind of sandstone, I believe, where it's within the bone itself, like it's all tucked away in a time capsule, basically, isolated
Yes.
from the environment. So
Exactly.
not even withstanding the environmental effects and just talking about the stability of the molecules, etc.
Yes, because the stability of the molecules is also going to be dependent upon like whatever elements they get exposed to.
Right.
Same goes for like ancient DNA. We can't really get ancient DNA from fossils that live along the equator. DNA will degrade at a faster rate the warmer the temperature. And so that's part of the reason why we are able to extract, DNA sequences from, areas that would have been very cold a very long time. And there was a recent study where they were analyzing DNA in the soil in Greenland. So they just took whatever DNA they could get in this frozen soil in Greenland, and it had the right conditions for kind of keeping DNA nice and stable. And they were pulling out, somehow they dated it to like over 1 million years old. And they're basically looking at genome sequences of all animals that were just shedding DNA, and it got into the soil and then frozen like 2 million years ago or 1.5 million years ago. And it included like all kinds of birds of prey, but also a bunch of animals you wouldn't necessarily find up in that northern of a latitude. At least they're not there today, but included like elephant-like animals, right? Mastodon and mammoth type animals, but ancient versions of these. This just came out like a month or so ago. It was really pretty cool. I saw an interview with the guy.
And in contrast to colder environments, especially, same reason why you don't typically find fossils from tropical rainforest type of environments.
Yep, because partly it's temperature, but also because those warmer environments and many of those environments in rainforests tend to be very acidic, which is very poor for fossilization. Yep.
So I was confused. TP Seeker, I saw on Restream, I could see his quotes, and I didn't see the paired. So how do you pair? Because it's sending it to your channel too, Chris.
Oh yes, I know. So for me, and he's showing up in the channel there. When I signed up for Restream, I must have clicked some kind of setting. So every time we do a Restream, it just shows up in mine. And I put, I said, you can see I said, hey, the conversation's being hosted on your channel. So I'm trying to put people over there, but TP showed up in mine, and yes.
Well, it's kind of cool, but I was trying to copy his links, and you can't copy out of Restream. It just highlights.
I can copy. I'll copy his link.
Well, I did it. I put him in.
Okay, you got it. Okay, great.
Once he had, yeah. Oh, I was so confused because I'm old and confused. Very confused. Anyway, so
do you still want to go over the other thing, speed, or?
I know you do. You just counted.
Of course, because it's about me. I'm just kidding.
You're counting. Yes. Me.
I feel like my ego hasn't done well over years.
Oh man.
Let's see.
So much for all of us.
YouTube.
Yeah.
Okay. So base theory clip 26 minutes. I'm not sure. Let's see. Do you come in here?
Yeah. Or do you want to go for both parts or just my part?
It's up to you. You can
if
do
it
anything.
involves Kent Hovind being destroyed, I am happy to see it.
I guess since it is from Grayson's video, maybe we'll just play my part and people can go watch the rest of it, or the rest of the discussion on Praises channel if they want.
Are you the red ball?
Yeah, that's my old avatar, remember?
Oh, no, I don't. I totally forgot it. Like
I start talking a little after Rage leaves.
Okay. So
So after that.
Is this a recent video, by the way?
Yeah, this was yesterday.
Okay.
Oh, yesterday? Okay. No, 3 hours ago. Grayson put it up 3 hours ago on base.
Yeah, he put up a few hours ago. But the original stream was yesterday.
let me put this in my description first before we even go there. But okay, so what part do you want me to play? Just play away?
I guess our yeah, because I start talking once Rage in the corner there left. So
Okay.
do you want to just
Did
if you want
Rage
to play
quit?
the whole thing, or if you want?
Yeah, let's do a sound check. Let me know if this is too loud or whatever.
Okay.
Okay. Oh, I'm muted. Gotta unmute the site. Okay, here we go.
Sea life that we find fossilized in the Grand Canyon. And
one topic at a time.
How is it? Is it too loud or loud enough?
It's a little loud. But
Little
what do you
loud.
guys think?
Yeah,
A little loud? Should I crank it down
But
a little bit?
just a bit. I wouldn't go down
Just a
too
little
much.
bit. Yeah.
Okay, let me know if we need to crank it up again. Okay, so we'll let's see. Let's go see where Rage leaves.
I did have a brief conversation with SFT after I was done with Kent, but we can do that another day.
Mhm.
I think that was the part I did less well on, to be honest.
It looks like Rage left right there. So let's roll it. from
Law.
that.
And it proves exactly what happened during Noah's flood. The tide going up and down makes the water going in and out. Hydrologic sorting would happen. Animals would be sorted based upon all kinds of factors:
Sorted.
habitat, mobility, body density, intelligence. So the layers all formed, I think, during the flood, and it probably took a year to make them all. So yes, animal tracks could be found between the layers.
Why?
What can you explain why there are?
And I'm just
One
going to
topic?
jump in here really quick. So we're going to let Grayson respond. This will be the last of that. Then we're going to head to Raging Atheist and then Rational Empiricist. But Donnie, SFT is here to help keep the
All right,
flow of
hang
the
on.
traffic.
Do you want me to go further ahead? Because Rage is still here. So
I don't know if he clipped out the part where Rage talks, so whenever Rage
No,
leaves.
he's got it. So let's see. Okay, looking. Oh my goodness! Oh, maybe he clipped out the part where you talk. And,
No, he has it there at the end section of it.
Okay. I still can't find it.
He actually he made me feel a little flattered that because he said he posted it for the ending too because of the part that I did. So thanks, Grayson, for reposting that.
So Rage is still here at that point, and then still here, and then we go to. Okay, is Rage still here? I don't think so. Okay, so here, let's try here.
By the angle of the crossbeds, we know this was.
I'll just end it there.
Okay.
I agree. You don't see how it can happen. I agree with that.
So now we'll hand it up to Raging. I know Rage has been waiting there. So go ahead.
What you see here is crossbedding, basically, and by the angle of the crossbeds, we know this was
Right.
formed by wind-blown sediments by a desert. And this is, like I said, just one example of these types of deposits. So how do you explain these massive sand deserts, basically, during a worldwide flood?
Well, again, it sounds like you're assuming the world was underwater the whole year. As I said before, the crust
You
of the
just
earth
said
would be
the
flexing
world
up
was
and down.
covered by water.
I do believe the earth was covered by water at some point, all of it eventually covered by water. Obviously, would you agree that these layers have evidence of some water being involved in depositing some of the layers?
No, they don't have any evidence in these specific layers, no. It's all wind-blown deposits.
Is it called sandstone or just a sand dune?
Oh my God.
It's a preserved sand dune that now forms sandstone.
Okay. How does sandstone form?
It can form many different ways. This particular sandstone formed in a sand dune environment.
Does a sand dune harden into stone ever?
Yeah.
Yes, we see that happening today. There, you can look underneath a sand dune and you can see the layers deposited over time.
I just swallowed my tongue.
But they don't form into stone and harden unless there's water involved. Is that a reasonable statement?
Okay, I would not pause consider for a that.
sec.
No, you
Okay.
just said
And
not all sediments can be.
Okay.
So I find it just a little amusing that Kent's this renowned geology expert on the panel here. doesn't know how sedimentary rocks form from sediment over time through pressure. And well, I think I mentioned later how there could be groundwater in the sediments, but that's not the same thing as like running water depositing and like where does he even get the idea that water needs to be involved, like flowing water, I'm guessing, needs to be involved for the sediments to turn into rock over time?
Yeah.
Like that doesn't make any sense.
Yeah, it could happen that, later on water seeps into that and starts depositing more cementing material and makes a different kind of sandstone.
Well, exactly what Brian said here. Sandstone from dunes and dry areas formed by heat and pressure from the layers above them. Yeah, exactly. Because if you try to imagine it, you have these giant sand dunes basically covering a wide area that are like hundreds of meters high or hundreds of feet high. And at some point, with enough sand over it, the bottom layers where this crossbend shown here exists is gonna gonna solidify into rock.
Yeah.
Yep.
Where the hell is it?
Yeah, and what?
And even like the idea that you could have this catastrophic flood, water, the way they describe it, like water gushing from underneath the mantle of the earth and falling all across the earth, that somehow you would have preservation of fine dunes that as lightly laid sand this deep underneath the rock, and that over time have been eroded away. How could you possibly have these layers of the sand be preserved in such fine layers, clearly indicating just like sort of a pattern of layer after layer being added over a long length of time and then being buried? How could that possibly happen in a catastrophic flood? And we see this over and over and over again. You would not see the lamination that we see if there was like a global flood. It's just impossible. And within different lamina, you have trace fossils. How can you have trace fossils, like animals moving around and doing their animal thing in a given layer, and then you look at another layer and you have a different group of animals doing a different thing?
Grayson mentioned that earlier in the discussion and kind of just waved it away with.
Blew over it. It's like, okay.
Some kind of know how to explain it.
Well, a tsunami came in, right, and deposited a whole bunch of wet mud, right? And then some animals ran across it as another tsunami was coming in, chasing them across the mud.
Yeah.
And then it killed them.
Except the problem is the trace fossils, some trace fossils that we know of, clearly is animals eating things along the sediment. And like you can identify this even in Precambrian fossils. So this is a little bit that I know about. Like in Precambrian fossils, you have Dickinsonian leaving impressions on these muddy sediments where they would have sat and then eaten and then moved a little bit, and then they sat and then they depressed down a little bit more. And it's the same animal moving 30 meters. They've tracked one of them out to 30 meters doing this in one layer. And then you go up another layer, then you have a completely different group of Dickinsonia and other Precambrian animals.
Or what about worm burrows? Like how the worms have time to burrow through the sand during a flood. As they were doing
catastrophic flood with a tsunami coming in.
I'm imagining this Wile E. Coyote thing. The Wile E. Coyote running across, runs
exactly.
and
it's so
and then going the other way, he goes back the other way, start on route another tsunami.
It was those environmental notes that you can make about a fossil and its surrounding that convinced me that there could not have been a Noah's flood. I mean, this was years before I became an atheist. I had a quote unquote bad habit of reading scientific magazines like the Smithsonian Journal and Scientific American and everything. And I would read about these fossil finds and how they knew because of this, and this was a slow, drifting, pool of water that it was in, and so things would settle slowly. Otherwise, they would look, more distressed or more, like a blender. And I thought that's true. Things look a certain way based on the state of the water. If is it a still pool Is it a
Right.
sea that's shallow?
Right.
And
Yeah,
everything.
it's the whole thing with sedimentology. You can look with what the environments look like today. Look what the
Yeah.
deposits look like in the rock record and connect the dots basically. So
Right.
So-
Like annual varves are a great example of that kind of layer of sedimentation that you can see.
So
But limestone requires calm, quiet water in order for it to form. It would be impossible to get limestone formation after a catastrophic flood. Like a catastrophic flood that then all of a sudden disappeared within a year.
Yeah, this is why holding creationists to account for their model or lack of a model, to where this thing they say needs to be consistent with this thing that they said before about maybe even something different, because this is all totally ad hoc from them.
right.
And that's what Kent Hovind has thrived on, is just making stuff up or memorizing stuff from other creationists and sounding confident. He's a confidence man rather than a man or a person of science.
So Captive Desk is going to play creationist geologist this summer with some
I look forward to that, Dave.
Yeah, we can't wait.
Nice. Yeah.
Gotta see that shit.
I've seen Kent Hovind speak in person. When I was in graduate school, he was doing I was pretty active in skeptic communities, and my thing was creation evolution, which is why I'm still passionate about it so many years later. And I would go to as many like creationist and intelligent design events as I could. I was a student at the University of Washington in Seattle, which is the home of the Discovery Institute. And that was a kind of an exciting time to be in Seattle because they were constantly hosting all kinds of stupid things. Of course, they never dabbled with like creationists like Kent Hovind, but there were also a bunch of younger creationists who would show up. So I've seen Dwayne Gish, and I've seen, but I've also seen Kent Hovind, and he was doing this like 3-day seminar at this, I think like either a Ukrainian or Russian church, an Orthodox church, and it was these long seminars. I went there with a friend, and we would sit there and listen to him, and he would go over like all of his stupid lectures, but including some of them where he was talking about like the end times are going to be upon us, that there's going to be a mark of the beast that's going to be put onto your forehead, and that mark of the beast is going to be a UPC symbol, like the barcode that you would get on the back of a box of Wheaties or something.
Yeah.
That they're going to put that mark of the beast on your forehead. They had a picture of that. And then he was talking about that we know about all of this, that there's this global conspiracy to trick people into believing in evolution so they deny God. And we know about this from the protocols of the elders of Zion. I shit you not. That
Wow.
Yep.
So you're saying that was Gish of Gish?
No, this was Kent Hovind who said that.
Yeah. Oh, that was Hovind. Okay.
That was Hovind. Yes. No, Gish didn't talk about. He wasn't a Pez dispenser at the event that I saw. No, but the protocols of the elders of Zion, he spoke about it favorably. And there was a time when he was selling it on his website. He took it off once. My friend and I, we wrote this up, and then it got picked up by the Southern Poverty Law Center, and they wrote a report about how there's this guy, Kent Hovind, running around talking fondly about the protocols of the elders of Zion, and that he was selling it on his website, and then he didn't sell it anymore.
Wow.
Yeah, that's who we're dealing with.
America thanks you, Dr. Thompson.
Yes.
You want to play a little more?
Yeah, it's from here to the end, I think.
When he says water, my brain comes to a full stop.
Same.
Okay, I wasn't going to mention this, but did he add a syllable to the word density? I swear he said density or something.
No, he goes density. The water. Oh, I hate the way he talks. It's so annoying.
So who the hell talks that way? What part of the country do people?
Okay, if you go to a carnival, you have somebody standing out there trying to get people to come to see the freak of the week or whatever on display. That he sounds like one of those, like a carnival barker.
Yeah.
Come and see the water.
Yes.
I think I would expect to see him trying to sell steak knives at Costco on a Saturday afternoon. And he also gives me the vibes of the mobile home trailer used car salesman
Yes.
on Sunday, Sunday afternoon or something.
What do I have to get you into this puppy? Very much.
We'll never get the Standing for Truth's Volkswagen sales pitches that we found on YouTube. I will never get that out of my head when I hear him talk. When I hear him selling creationism, he has exactly the same style as when he was talking about the strength of the frame on the Volkswagen and all this bullshit. It was amazing. It's amazing.
Yep.
Anyway, okay. I'm gonna roll. Shall I roll?
Okay.
We're rolling.
Need water to be deposited, correct?
No, I'm asking you, how does a sand dune turn? Can a sand dune turn to stone with no water involved whatsoever? You're saying a pile of sand. Okay, we have sand dunes right here on our property. Come see Dinosaur Adventureland. It was a gravel pit. There are giant sand dunes here. And sure enough, the wind blows them around, makes layers just like this. They will not turn to stone, though. There has to be a chemical reaction, and it involves water.
Holy
so
shit.
I think what you're showing is clear evidence of Noah's flood. I've been there to the Navajo sandstone. And
I just realized this. Hold on.
Oh, wait a minute.
He thinks water creates rock by a chemical reaction.
Mm-hmm.
I don't know. But
yep, that's what he's saying.
Okay, so he's saying that he's saying that after the flood, it was dry, and then all these sand dunes formed, and then another flood came and turned it into rock. I mean, wouldn't it have just blown it away if it was a sand dune? I mean, what?
I think he's just trying to recall it from his readings of Henry Morris, about the flood. I forget the name of his book, but
I got the book somewhere. I should look at that and see how he says these things were formed. But
and Brian Stevens makes a very good point here. Yes, the sand pit is larger than the dune sand. Yes, that's a very good difference there in terms of geology.
Yeah,
like very coarse sand and coarse gravel are not the same thing.
I can't believe he said that. I mean, either he is not very smart.
I'm not sure I understand.
Oh, shut up, Siri. Either he's not very smart, or he knows that the audience he's preaching to are not going to be critical of anything he comes
pretty
up
much.
with. I mean, my goodness.
He's just under the impression. I mean, it's true that water passing through sand deposits, you're going to have the accumulation of clays, smaller grains, those kinds of things that will allow for greater compaction once that sand deposit gets laid upon more and more sand. The pressure then is going to allow for the formation of sandstone, but it isn't absolutely necessary. You don't have to have water allowing for that compression to occur. Weight is needed and time is needed. That's the key.
Exactly. Yeah.
Water can help.
Yeah,
groundwater, and you're going to get different kinds of sandstones
together.
Yes. Right.
Oh, has everybody caught the 4-part series? I think 3 parts are out so far on James Tour.
Part 4's out too.
It is.
It's priceless.
Oh, it is good. He just shatters him.
At the end of part 4, I don't know that this is a spoiler, but he actually does challenge James Tour to a debate in Houston in May.
Good.
And he does have a couple of terms, like the moderator has to be a non-creationist. But he's offered that challenge, and he said that James Tour personally must respond, not any of his handlers at Discovery Institute.
Yeah. So, Chris, did you see the part where James Tour was talking about finding red blood cells in Tyrannosaurus Rex, and musing that they might still be alive somewhere on the Earth?
No, I think I missed that. He said that.
Dr. Joel Duff did a video on that.
Okay.
Yeah.
So James Tour, is he a young Earth creationist then?
I guess so. He's kind of coming out of the closet.
Yeah,
Like he
I
is.
guess
Pretty close.
that's
He
shocking
alternates.
because he's like, a Discovery Institute guy, and like they're not
Yeah.
supposed to talk about stuff like that.
Well, okay. Dr. Joel Duff does a really good. He shows some of James Tour's blog, or is that Professor Dave? One of them show the blog. That's Professor Dave. They show the blog where James Tour talks about his religion, and he sounds a hell of a lot more like a creationist fundamentalist in his blog.
I mean, they all are, I'm sure. I mean, all of them are to a good degree. It's just that the ones who have some sort of professional credentials and have ascribed to the intelligent design model, they take the stance that our job is to try to poke holes in evolution as much as possible and talk as little about our own model of intelligent design. We don't talk about our model at all. It's just from inference. If we can disprove A, therefore B must be true. And that we only argue from an argument from ignorance and from incredulity, and that's it. And that is the intelligent design model. So they're not supposed to talk about stuff like, oh, dinosaurs should also still be around today, blah, blah, blah. Well, dinosaurs were on the ark. Okay.
Yeah. We're going to find them somewhere in the Amazon.
Yes.
After watching that documentary, King Kong Skull Island, I think that convinced him.
Somebody's dog just saw a Tyrannosaurus Rex.
Yeah, that was my dog telling me, go sit on the couch, because she insists that I sit on the couch around 4 o'clock in the afternoon for some reason.
Pet time. Yeah. Yep. Okay, I'm going to play more. We got. I think we got about 3 minutes, 3.5 minutes left. Okay.
I think during the flood of Noah, the tide going up and down and the crust of the earth flexing up and down is certainly capable of creating this. Now, if you believe in evolution, I'll ask you a slightly off-topic question. Do you believe all these sandstone layers fit in a dot smaller than a proton eventually, way back billions?
That's not what we're talking about, Ken. So please.
Okay, I know. I know you don't want to talk about that, but if you believe in evolution, that is what you believe. All those grains.
Well,
of sand,
he's a dick. B, talk about changing your subject.
Big time.
Yeah, and that has not, the one has nothing to do with the other. If you're an evolutionist, you got to believe in the Big Bang. Well, no, you don't.
Not necessarily.
Okay.
Nope, you don't. I put in the private chat one more thing, going back to collagen to kind of bring it all back. This is a review article talking about the role of collagen in wiring the vertebrate nervous system.
Oh, good.
Yep. So this is also something, and it turns out that this is the director of the School of Neuroscience at Virginia Tech. This is when he was at VCU, but he's been hired here to be the head of the neuroscience here. His name is Michael Fox, not Michael J. Fox. He's Michael A. Fox.
Nice.
Yeah. But yeah, he's a great scientist. He does really great work. And this was some of the work he was doing about 10, 12 years ago on how collagens are secreted from cells, and they act as signaling mechanisms to allow axons to wire up in the brain and get connected to the right places. So they don't do any of that kind of function of extracellular, just sort of cushioning and protecting or rooting cells. They just act as a signaling mechanism. So there are collagen receptors that are sensitive to this family of collagens that are excreted from cells, allowing the axon to now go down the gradient towards the collagen source and then connect to the neurons there. That's how it works. Completely different than like all the other collagens.
Yeah. Yeah, and Andrew, I want to talk to you at some point about morphogenesis and
yeah, sure.
Basically developmental biology, because God is, and we have to start talking about is all the things that cells can do to each other, and what the local environment of every cell is. And it's there's just a whole lot of that of really cool stuff, and that's one of them that you mentioned, Chris.
Yep.
I'm putting things in my description here. I'm putting these papers in. And I shall save that. Okay.
Anyway.
Move ahead.
That's why you don't show the face. I understand. Go ahead.
Ken, are you suggesting that these sand dune deposits, basically. Because if you look at the reason we know, let me clarify this, the reason we know that they're sand dune deposits is, like I said, because of the angle of these cross beds, because sand has what's called a maximum angle of repose because of the dune slumping over itself, basically. That's how we know this is a sand dune preserve, basically. And in my view, at least, and in the view of the vast majority of geologists, this is inconsistent with the world being covered by water. It certainly couldn't be a dune migrating and slumping over itself during a worldwide flood.
Well, I think you'd probably have to talk to some of the. There are some creationists who specialize more in these features like this than I do. I'm looking at the much bigger picture of, you believe all these sand dunes of the whole world, all the stars fit in a dot. You want to try to find fault with Noah's flood with something like this and say, because.
See, he got embarrassed, and he knows.
Of course, yeah.
And now
he's trying to bring up the Big Bang.
Right.
Oh my God.
This flood story doesn't answer this question. Therefore, we're related to mosquitoes, and we all came from a dot. That's the logical conclusion you guys come to.
No,
you believe in evolution?
I bring this up because it's fundamentally in conflict with the idea of a worldwide flood.
Well,
And
I don't think.
This is
well, let me address that real quick. I'm co-host here, but I do want to point out that this question about the sandstone here, the Coconino sandstone, whether it's deposited in a dry desert or during the Genesis flood, you guys in the secular community say that it was a desert, but there have been an entire team of geologists, PhD geologists, in the field who went and looked at these supposed 7 facts that support this desert hypothesis, Andrew, and they actually discovered that they each match the flood interpretation perfectly. So you saying it's in complete...
I wasn't discussing the Coconino, though. I was discussing the Navajo and several others.
Oh, that's my point, Donnie. They look at one thing like this that they think creationists can't explain yet, and therefore conclude that they came from a rock, which came from a...
That's exactly what they do with evolution. Yeah. I mean, he just described there's one thing that we don't know about evolution, and God damn it. Yeah. Anyway.
No, and then he just, I was going to say, he paints this caricature, this silly caricature, that you evolved from a mosquito and you came from a dot and you grew from a rock. And it's like, dude, you can't answer this one question because it's completely incongruent with your model. Clearly, you laid out a nice case showing like this. And you could point to a lot of other examples, but you showed a really great example.
This was just one of them.
Yeah, but there's like, clearly he doesn't have a response. So then he just, takes instead of trying to answer it, he just comes up with a completely ludicrous caricature to try to make the evolution side look ridiculous.
And
and not deal with the actual issue.
As Grayson mentioned in the description of this video, actually kind of amusing how SFT came to rescue Kent, so to speak. But I will comment because I wasn't able to in the actual stream, but the stuff SFT brings up, actually other geologists have looked at those creation geologists misrepresentations, basically, or strawman of how sand dune deposits actually work. For example, I'll give you just one of them. Going back to the angle of repose, you'll find some creationist sources which say, oh, the Coconino has an angle of repose of 25 degrees on it. And I'll say, what the angle of repose of sand dunes today is up to 30 to 34 degrees, but they won't tell you conveniently that the range is actually from 10 degrees to 34 degrees or so. And the average is, surprise, surprise, 25 to 28 degrees or so. And in fact, water deposited cross beds rarely get above 10 degrees. So just a little bit on how those studies fare.
Yep, that's interesting.
Well, you believe, Andrew, that you're related to water.
And that you both fit snugly into a dot.
Came from a dot. And you were a mosquito, and then you came from the mosquito, and which came from a rock. And that's what you believe.
Well, I think you need to enter the the Kent Hovind impersonation.
Oh yeah, was that
Olympics.
pretty good? Oh God, he's so irritating.
Well, this was my my second official interaction on air with Kent Hovind, so I'd love to hear y'all's thoughts, and constructive criticism, of course, is welcome. But I think I did a lot better than last time. But I know I
You
can
did really well,
do-
but you know you'll never get that time in your life back.
That is
Yeah,
true.
that's true.
Yeah, and
Yeah.
there's a lot more to this video where Grayson came in and started talking.
Oh yeah, Grayson did good too. Yeah, before I knew.
I linked it in the description. I suggest everybody watches this.
Thank
It's
you.
pretty good.
I've been asked to debate Ken Hovind, and I have no desire. He's too slippery. He's too
It would be boring.
sarcastic and yes, and disrespectful, and he doesn't argue anything in good faith. He's never ever going to say that you have anything like of value to say, and he's been told he's been wrong over and over and over again, and he will never ever admit to being wrong, or that like his idea could potentially even be wrong. And there's just no point in getting involved with that. I mean, it would be a complete mess.
When Kent actually challenged me to debate him on SFT's channel on the actual stream just now, I was very tempted to respond with something along the lines of, I would rather go stick my head in an oven and put my feet on hot coals for a while.
That's so funny. I said essentially the same thing. SFT asked if I wanted to debate Ken Hovind on his channel, and I said I'd rather gouge my eyes out with spoons than to debate Ken Hovind.
That's pretty good. Yeah.
Now, I like what Jerry Coyne always says when he's challenged by people that he just really doesn't think much of. He'll say something like that would enhance your CV while it would degrade mine.
Degrade yours.
Oh yeah,
Right.
Yeah.
I feel the same way especially with Ken Hovind. I mean, he's just a clown, and he's ridiculous in so many ways. And the fact that SFT has hitched his wagon to him, most creationists, including lots and lots of young Earth creationists, have totally sworn him off because he uses complete garbage arguments over and over again that have been disproven that even other young Earth creationists won't use. I mean, he's a pariah, and it's just goofy that like.
Not to mention all the other slippery and domestic stuff attributed to him. So.
Yes, there's that too. Yeah. All right, all that stuff. I mean, I don't necessarily want to attack personal character, but he's certainly someone who has demonstrated over and over again that he lacks it. So,
yeah,
and there's no point in engaging in someone like that. So, like other young Earth creationists, I don't have a lot of respect for like other young Earth creationists, but I at least respect them as people. So I don't mind engaging with them, but him, it's a completely different story. And that when I saw him talk about the protocols of the Elders of Zion and praising it and saying that somehow this is like true and that we should be learning from the protocols of the Elders of Zion, it's like, God, no way.
Yeah. So
I definitely don't need to elevate someone like that.
I swear he thinks all these things are movies or documentaries because, there was King Kong and Skull Island. There was all the Dan Brown movies with Tom Hanks.
Yep. He's got a lot of weird ideas beyond just the young Earth creationism.
So Brian Stevens, are you a geologist? Sure seem like one.
He seems pretty knowledgeable about geology.
Yeah,
what are you, Brian Stevens? We want to know.
Email me. Yeah, what are your credentials?
He is Brian Stevens, nothing more.
Brian Stevens, nothing more. Okay.
It'd be useful to have, because I can talk a little bit about fossil formation, mineralization, but I am a neuroscientist. I am speaking from an avid amateur at best, and one who has read up on these things. I'm definitely no expert, that's for sure.
I'd love to see what Jorge's opinion on if he were to review the discussion, see what he thinks.
So I started getting back into neuroscience, Chris. In the last few weeks, and
I've seen, on your streams you've talked about the brain a few times.
I have forgotten so much, and the default mode network came after all the textbooks that I have, and some of the networks are talking about. And there's also a new article about layer 5 cortical neurons by Jeff Hawkins. Are you familiar with his AI stuff?
No.
Okay, Jeff Hawkins, he wrote some kind of a video game, I think, or something, or so. He made a lot of money, and then he went into studying consciousness. And he has a couple books on theories of consciousness, and they are really detailed as to what the layers of the cortex do, how they interact, and how they predictively light up the next neuron in the pathway. And just wonderful. I've got a wonderful book on them that I have to review again. But it's been almost 10 years since I did neuroanatomy and all of that. And I have forgotten so much. It's horrifying.
So he's got a book called 1,000 Brains, A New Theory of Intelligence.
Yeah.
He does? And that came out in 2021.
Yeah, he's got some older ones.
Those older ones. Right, On Intelligence is like I think one that's a really well-known one. But he developed the Palm Pilot and was also involved with Handspring. So that's what he did before he just got into neuroscience, like in the early 2000s, which is kind of crazy.
So Brian Stephen is autodidact from hell. That's it.
We are all autodidacts here, except for Dr. Thompson.
Yeah, we love autodidacts, especially from hell. Satanic autodidacts. We love them here.
So is the auto? Does that mean like financing? Because if you voluntarily read, I mean, Dr. Chris Thompson, he read a lot and studied a lot and did experiments. But he just had to pay for it, whereas Speed and Andrew are both reading and doing a lot, and they're not paying for it. They're quote unquote financing it themselves.
Right. Like in 2011 or 2019, I spent $20,000 on books.
Wow.
I'm sure you look up a lot of stuff on your free time too, don't you, Dr. Thompson?
Oh, for sure. Yeah, and all this stuff. I mean, even though I have an undergraduate degree in evolutionary biology, most of what I learned is on my own. And I mean, and I don't know. In some ways, like, of course, I learn more and more about neuroscience. No one's teaching me it. I have to go out and read it and study it my own. I'm not taking classes anymore, but I'm still learning. So in many ways, like, professors are very autodidactic.
Yep.
It's almost like that's a skill you have to include in your life.
Absolutely.
To really ever qualify.
Yep.
So basically, we just need to get some PhDs to quiz Speed and Andrew and see if they're at the right level, and then do like some sort of thesis or experiment.
There you go. Yes, you can do a defense of what you've learned, and
I run away every time I'm tested.
You don't need the credential. It doesn't. It's I get to call myself doctor. That's great, but it's like, okay, it's all about the individual.
The last college classes I took, I did them with basically audited them, basically a pure audit. So I got no grade. I had to take no tests. And because I did it that way, I think I absorbed more from those classes than I ever could have if I had played to the tests. And it was amazing. So I will never take a course for credit again, because it screws up your whole, it puts you into short-term mode instead of long-term and overarching theory mode, right?
Yeah.
I took some math classes and I just basically did a pure audit, which costs like $12 a credit in Minnesota for seniors, which is pretty cool. But I got so much more out of those classes than I did the ones where I tried to get a grade. And that's
kind of reminds me of, or makes me think about the older days. Let's just say when a few centuries ago, no one knew all this stuff about geology or biology. Everyone had to, or these people had to go out into the world and figure it out for themselves.
Yeah. Yep. What do they call it? The savings and loan education plan, where you basically you learn the stuff and then you pay it back. You give it up on the test and then you forget it. So I don't know. That's just
Well, I think
personal
I
experience.
think our model of education is really kind of whack because like in your lecture halls, you have these huge groups of people that can students that can really in no way interact with their teacher. Maybe the first 5 or 6 people who run up to the teacher as soon as class is over might get some personalized back and forth. Even going to the office, everybody
Office
won't
hours.
do that,
Yeah.
and everybody can't do that.
That's right.
So how are you really going to get challenged by a back and forth, or and discussion, or even pop quizzes administered by a teaching assistant or something like that? I don't even know if they do those at all in college anymore.
Sure. Yeah. I'll give pop quizzes to my students.
Because you need to, number one, you need to stress a student's brain, and also you need the feedback to know if they're really engaging with material. And the question is that how much do professors and educators care that their students are engaging with the material?
I think, okay, so I can speak from experience here. I think in general, at least my experience is that the majority of them do care, but not a huge majority. There's certainly plenty of professors who, I mean, I guess they care about teaching material, but they're also like, well, it's more sink or swim. If you can't get it, too bad, that's on you. Some students do get it, so obviously you could get it if you put in enough work. I think most professors, though, actually do care about that kind of feedback, getting it in the middle of the semester, and trying to adjust their teaching styles so that students are actually understanding it. Because, like, what's the point of what we're doing? We do want to teach our students. We want them to learn this material. And if they can't get it, well, then that's not really on them. That's more on me. I got to get down to whatever their level is at, figure out what that is, and teach them to that level, and then get them up. Obviously, if I've got a class with 100 students, and I do teach a class with that many students, I can't get every student. That's just going to be impossible. But I try to target like the lower third, and also leaving enough stuff for the people who are up at the top to also get things out of the class and appreciate like what they're learning. Students vary enormously in how prepared they are for certain classes.
I've hardly ever found a teacher that when I went and asked them deep questions or the right questions that they didn't immediately sit down and really accommodate me.
Yeah,
I mean,
right.
But if you go and ask stupid shit or you don't put it to work, they can blow you off, and they should, as they should. Yeah.
And I think most professors will take the time to answer a question, or, and oftentimes I'll get a good question from a student and I don't know the answer to it. I don't know everything about neuroscience by far. So I look it up, and then I might bring it up in class the next time, or I reach out to that student and say, hey, I found this paper. This answers your question. I do that kind of thing. A lot of professors do.
Cool. Indeed.
Yeah, so I'm getting back into neuroscience at the same time that I'm doing a deep dive into my own brain and life, and I'm trying to put things together with things like the default mode network and stuff like that, and it is really interesting. Yeah, and I've always done that. I've always really looked into subjective experience as basically the thing I want to explain, and try to understand what is it, like, how can I grasp what my own subjective experience is in different ways? And then how can I explain that with the neuroscience, which is pretty cool. So.
To get people up to speed on the default mode network, what that is that it's these series of brain areas that tend to be quite elevated in activity when a human being is really not doing anything that requires specific attention, or if they're working on something. When they start to work on something, activity in these areas tend to go down, and then other areas seem to get active. And the idea is that the default mode network might be that kind of wandering conversation that's happening when you're just kind of thinking about things or puzzling things through. Because there are times when the default mode network goes up when you're doing some sort of attention task, but you got to think through a problem. And now all of a sudden the default mode network goes up. But if you're doing a task and you're doing what's called exploitive, where you're just following the rules. You know the rules. You're just doing the task. Okay, I click this. I click that. I click this. I click that. I know the rules. I'm getting good feedback. The default mode network goes down and attention centers are up. But as soon as the rules start to change on you, that's when the default mode network goes up again. It's a super fascinating phenomenon. And
there are some contradictions in all of that I'm finding. And
right.
That's why I want to look more into it.
And the default mode network, we call it the default mode network because we would put human beings into scanners and we'd say, okay, don't think of anything, and because we want to get a baseline activity of the brain areas. And then there'd be areas of the brain that would be firing and be like, no, you must be thinking of something because we have these brain areas that are firing. So don't think of anything. I'm like, I'm not doing anything. I'm just sitting here. And they call that the default mode network because they tend to be active when we're not really doing anything. And these are the areas that tend to be the most energy hogs of the brain. They use the most energy in the brain.
Yeah.
Super cool.
Yeah, I mean, I'm interested in the maze of beliefs that we have about the world, politics, everything, and how those things are basically, they seem like they're kind of the default mode, that we don't do a lot of thinking. Like if somebody asks you, do you believe in X? You'll knee-jerk respond, yes, I do. Yeah, I do, without really doing a whole lot of thinking about why you believe in that.
Right.
And those things really interest me. And then when you start thinking about why you believe in it and you start digging around and trying to pull those things out, it would be interesting to see that process scanned.
For sure.
Like somebody's in the default mode and you ask them a question and they answer immediately, yeah, okay, I believe in that. And then you ask them to start digging around and explain, well, when did you start believing it? Why do you believe it? What are the things that led up to it? And I'd like to see the interplay, because that's kind of what I'm doing, is trying to take my belief system apart, right? And look at the sources of it. And a lot of those beliefs are about who I am, what kind of a person I am, and what kind of a person I've been in my life. And what I think other people think of me and things like that. And I'm just taking all of that apart.
Sure.
So I'm in kind of an intensive therapy with a gentleman who I really like, and we're going deep. It's pretty cool. So
cool. It's great.
Having fun. Back to neuroscience. I needed to get back to neuroscience.
It's a great topic.
My one comment about that is when somebody asks you, do you believe in evolution, or do you believe in God, or do you believe in life after death? To me, we by default assume that we both know what that word means, and instead we should probably say, what are you asking me, really?
Mm-hmm.
Yeah.
And kind of put it back on them to use less generalized, less ambiguous words to ask me the question.
Yes. And I think, because some of it, they want to trap you into it's like faith or something because you just believe it. And my response usually is, well, I don't believe it per se. I accept that evolution has overwhelming evidence that supports it as a working model. And then, of course, if new evidence comes about that would not be consistent with that model, I'm more than willing to change the model. I just haven't seen it. Yeah. Of course, that's a lot of words. I like your idea of being like, well, what do you mean?
Yeah, that might get you invited to fewer parties, but then again, those are probably the parties you don't want to go to anyway.
That's right.
Because in a lot of ways, our words kind of signal to other people where we fit in the social order, what kind of person we are, and so on and so forth. Because they could be really asking, are you an okay person to talk to? Are you in my social and economic class?
Are you going to kill me?
So what?
I always want to know if the other person's going to kill me or not.
Yeah, are you going to kill me? I think that's probably the best pickup line possible. Are you going to kill me?
Yeah. Yep.
Are you just going to make me wish I'm dead later?
And then she says, I want to. But, yeah, there you go. Okay, guys.
Yeah, I got to get running.
Yeah, let's wrap it up. I don't have an outro, do I? Or
Well,
do
this
I?
was an excellent conversation, Dr.
Yeah,
was.
Chris.
thanks
I
for
do
talking.
appreciate you
Yeah,
being
it's
here.
been fun. For sure. Nice job, Andrew, on that.
Thanks.
On what you brought up.
Thank-
You handled that really, really well, I would say.
Thank you. I appreciate
Yeah.
that.
Thank you for showing up, Andrew and Chris.
Yep, sure thing.
Yep. Scott always shows up, so I should
Yeah,
thank Scott.
it's hard to get rid of me.
Doesn't get things at this point.
Speed even changed platforms to try to get rid of me, and I still show up.
I know. Yeah.
You do
And
good, Scott.
Yeah, but in my defense, I always show up too. So
Yeah, there is that.
Yeah. So thank everyone, including me. Okay,
Thank you,
guys.
everyone, for watching.
Shoot her in the head. Thanks everyone out there in chat.